Hi, Neto here. Today we're going to go over cellular respiration, or in other words, where does all the good stuff go? So we've been going over a lot about where the waste products go, especially in our poop lab, but now it's time to focus on what about the nutrients that we do keep and is absorbed by the small intestines? Where does that end up going? A good place to review is visiting our science webpage, and I would really encourage that you watch the other two videos that are on the science webpage to give you a full comprehensive look at everything that we're talking about. There's quite a few coenzymes and um, breakdowns of different molecules that we will not be covering in this um, debriefing of cellular respiration. However, they do go into it further at the other two videos. So this video specifically is just to give you a broad overview to hopefully help piece together some of the complicated topics. Cellular respiration is one of the most complex topics that you will learn in biology. So again, I'm really kind of trying to piece it down or break it down so it makes sense to you guys. And hopefully at the end of this video, you would be able to discuss these 5 questions comfortably, really focusing on understanding that formula, the 3 steps of cellular respiration, and what is ATP. What does your body do with all the extra sugar? Well, really, that's what cellular respiration is all about. It's about what your body does with sugar. So the main purpose is to make ATP. However, any kind of leftover sugar will be either stored as fat or in the liver. And then if your pancreas and liver are just exhausted from storing it all, that's when diabetes type 2 comes along and it can end up in the bloodstream, which can be dangerous. So far, you've learned about how the body transfers oxygen to cells and how the body transfers sugars to cells. But just to recap, the transporters of these molecules is insulin and hemoglobin. So I want you to think of insulin and hemoglobin as your car, and the passengers are glucose and oxygen. And so, in the process of starting cellular respiration, it's really important that you realize that you can't start it without these 2 major molecules, glucose and oxygen. And they won't get to the cells unless they have insulin and hemoglobin to help them. But now that the oxygen and sugar are at the cells, what do we do now? Well, that is, in fact, the next step. That's cellular respiration. And the goal of cellular respiration is to take sugar and oxygen and turn it into ATP. As you may recognize, the first part of this is the first part of your equation, which is the reactants section of the equation. And what is ATP? All cells need energy, and the unit of energy that cells use that's functional for a cell is ATP, and it's adenosine triphosphate, or ATP. The triphosphate is the key thing to pick up here, because we will also be seeing a lot of diphosphate, di being 2, tri being 3. So you would look at 3 phosphate groups in this one. So where does cellular respiration actually occur? It occurs in 2 organelles, and you want to remember this. The first organelle that is involved in cellular respiration is the cytoplasm. That's the liquid that fills the cells. And the second organelle that's involved is the mitochondria. And that really is called the powerhouse because that's where a majority of the ATP is made. And really, there's only 3 steps to cellular respiration, and you do need to know this. If you don't walk away with anything else, these are the 3 things that you need to memorize. Glycolysis, citric acid cycle, or Krebs cycle. So I want you to remember both names, but we'll be referring to it as the Krebs cycle here. And the electron transport chain. So our first step, glycolysis, and pay attention to the word, break it down, but lysis, L-Y-S-I-S, is really the breaking down of something. And glyco, being glucose, or glycogen, is really just sugar. And so our initial step is breaking down the sugar. So sugar is just too big for our cells to ingest. And so if you notice the chemical makeup, Sugar is a 6-carbon ring, and in order for our body to ingest it, or our cells to ingest that 6-carbon ring, needs to be broken down to 2 3-carbon chains, and those 3-carbon chains are called pyruvate. And so in order for this process to happen, it actually takes a little energy. And so the ATP is broken down or used to break down the glucose. And in that process, it loses a phosphate. So the ATP turns to ADP. And out of that process, you get 2 pyruvate and 4 ATP. We saw that in our demo with the sugar molecule being broken down in the cytoplasm, and we actually used 2 ATP in order to get that process. And again, this is a brief summary, but the main thing I want you to look at is that ATP and pyruvate are the output of glycolysis, and that's necessary for the next step, which is the Krebs cycle. So onto the Krebs cycle, or the citric acid cycle. I want you guys to focus on the word cycle because it's a constant cycle. And so going into the Krebs cycle is the pyruvate. And coming out of the Krebs cycle is our first byproduct that we see, and that's CO2. So all the CO2 that we're breathing out, that's from the waste product of the Krebs cycle. That's really important. Another byproduct or output of the Krebs cycle is the CoA, that's the coacetate, and then also hydrogen and ATP. In our demo, we used that our hydrogen was really the chocolate, and our NAD plus was our transporter. So those were the two things I wanted you to pick out from that demonstration, was that the NAD plus was actually picking up hydrogen and transporting it into the electron transport chain. Again, you needed to have that constant cycling in order for that to actually happen, and it took in ATP in order to have that process happen as well. So again, the main things that we want you to know is that it occurs in the mitochondria. The input from the glycolysis is actually what makes the Krebs cycle work. The output is CO2, hydrogen ions, and ATP. The Krebs cycle is aerobic, so it needs oxygen in order to work. And then it has this coenzyme NAD plus, and that is a transporter for hydrogen ions, and that's similar to hemoglobin and insulin. So our ladle, or our NAD plus, is taking all those hydrogens and turning it into NADH and actually taking it onto the electron transport chain. And this is our last stop, and it's a factory line of ATP synthase and synthesis, and it really is. It's just like a factory line, just outputting a lot of ATP all at once. And in our demonstration, I want you to recognize that we're using the muffin tins as a representation of the inner mitochondrial membrane. Which, if you remember earlier when I was talking about the mitochondria, it's actually very squiggly on purpose. So there's to actually increase the surface area of the mitochondria, and that all that squiggly line within that organelle is lined up with electron transport chains. And so throughout that entire squiggly membrane, we are getting ATP being produced at a rapid rate. Here you see your NADH being transported into our muffin tins, and then the proton gradient. I think this was a really cool analogy. Just like you wouldn't cook one muffin at a time, you're going to fill up the muffin tin and then put it into the oven. The proton gradient works the same way. It's a collection of the hydrogen ions on the inner membrane that is actually going to work in order to then push through the hydrogen ions through the ATP synthase. Um, so you won't see one hydrogen going in at a time. It's really that proton gradient. It's that collection of hydrogen ions that's actually forcing that current of hydrogen down and through the enzyme. And that last one is the ATP synthase photo, which is our enzyme. And if you haven't reviewed enzymes, if you're still feeling confused about enzymes, you need to come see me or review the videos or look on YouTube, because you're not going to get away from enzymes. But I love the oven enzyme analogy, because really what it does is it speeds up the reaction. And just like with these muffins, we're going to put those muffins in. They're going to actually bind. The hydrogen's going to bind with ADP, and we're going to get ATP. So it's going to speed up that reaction. The main things to know about the ATC is that it occurs in the inner mitochondrial membrane. Input of the ACT came from the Krebs cycle, and it makes a lot of ATP, about 24 to 32. It needs oxygen in order to work. It has this ATP synthase enzyme that is made out of proteins. All enzymes are made out of proteins. And that takes that hydrogen ion and the adenosine diphosphate to make ATP. The hydrogen ion, it will pass through the proton gradient. So again, you're going to see a collection of all those hydrogens on top, and that collection is actually going to force it down within the enzyme. And the byproduct of the electron transport chain is water, which you will eventually either sweat or pee out. I love this culminating kind of oversimplified explanation of cellular respiration, and it really talks about, it focuses on what would happen with oxygen without oxygen. And so it's really important to understand that we need oxygen in order to get the maximum amount of ATP out of a sugar molecule, because without oxygen, you only end up getting 2 ATP, and you end up producing lactic acid if you're an animal, or alcohol if you're a plant. So the burning feeling in your calves when we had you working out last week with Diane Wagner's was the lactic acid buildup, and that's because you're not getting enough oxygen to those cells. So one more review. Hopefully you're understanding this equation that much better, but your input is going to be the glucose and oxygen, which you're getting from your food and breathing, and then you're going to have a byproduct of carbon dioxide and water. That's your waste. And then what you're walking away with is a lot of energy, ATP. And that, in a nutshell, is cellular respiration. Let me know if you have any questions. Watch the other videos and try discussing those discussion questions as a good way of reviewing what you know. Have a good weekend.